sit over there.
Yes, thank you, Dan. Thank you very much for a nice and long introduction. Good morning, everyone. Welcome back, the third day of the conference, and I appreciate the speakers, being here, and those of us that are not speaking also being here. No, thank you all. So I presented the first day a concept where we used to induce tolerance to pancreatic islets for the treatment of type 1 diabetes. And if you remember, the concept was displaying on the surface of islets an immunomodulatory molecule, Fas ligand, and simply using pancreatic islets that engineered with Fas ligand to overcome allograft rejection. And if you remember, this is a pretty practical and versatile concept. It takes about 2 to 3 hours to engineer tissues and cells with Fas ligand. And then, as I indicated, islets engineered with Fas ligand will overcome rejection not only in allo as well as in xeno setting. We've also demonstrated that they will overcome rejection in pre-sensitized recipients. And if you remember the concept that, we take simply the idea is that to put Fas ligand on the surface of donor cells and tissues. We've done it for cells, tissues, even complete organ. When you transplant engineered Fas ligand, engineered tissue into a recipient, the T cells, alloreactive T cells, targeting the tissue will get activated, upregulate Fas on the surface, and Fas will engage Fas ligand on the surface of engineered tissues, and this will induce apoptosis in pathogenic T cells. Instead of T cells eliminating the graft actually does the opposite. Graft eliminated T cells. And this is called activation induced cell death. And this is basically the premise of the work. And we wanted to provide a proof of concept whether that indeed happens in vivo. And we used basically a mouse model that expressed of albumin on the insulin promoter. This is beta cell, they express of albumin. We engineered with Fas ligand, put into 57 black 6, and then 5 days later we took OT1 cells, the CD8 cells that respond to of albumin, that labeled with CFSE, and adaptively transfer into the recipient. So the idea to track these T cells and to see what happens. So these are alloreactive T cells by definition. So this is Fas ligand engineered islets. If you notice, we look at kidney draining lymph node where we expected the action. You don't really see that many cells, perforating. And we think the cells undergo perforation and they get eliminated through the apoptotic process because that's such an effective process. And apoptotic bodies are cleared off the system right away. We think that's the reason we don't see it. We went ahead, I'm not going to show the data. In vitro, we actually demonstrated indeed that's the case. And if you notice, in control animals where islets were engineered with controlled streptoavidin, in that case, you see beautiful perforation, especially in the kidney draining lymph node of those alloreactive T cells. You don't see much in mesenteric lymph node or in the spleen. There is some level of perforation. So the action happens locally. So this is to demonstrate that indeed Fas ligand engineered allograft, they eliminated tissue. So the next question we asked, because this is a localized immunomodulation, we put Fas ligand on the surface of the islets, we asked the question, does the tolerance we observe, is it systemic or is it actually localized? To answer that question, we performed this in vivo mixed lymphocyte reaction. And if you're not familiar, what we did, we took splenocytes from long-term animals. These are the animals they have accepted the graft for long-term. We labeled them with CFSE, then we injected those into an F1, irradiated F1. So meaning that the F1 will not respond to donor cells, but donor cell will recognize allogeneic coming from biopsy and will respond. So it's one-way reaction. And then this is a third party. Basically, we inject cells into third party as well as allo. This is the donor. And you notice the cells actually respond to third party as well as allo. And this was irrespective of where we harvested the cell from. So demonstrating that indeed in the periphery of the animals they have long-term graft survival. In their periphery, they carry alloreactive cells. They are capable of responding to the donor, but yet those cells do not reject islets. And if you remember, this is the slide I showed on the first day of the conference. If we asked the question, is it possible that because we have a normal alloreactive response, maybe there is a localized immunomodulation that prevents graft rejection, and the most possible candidate are FOXP3 T regulatory cells. When we depleted those cells either at day 100, all the animals they rejected, day 15, all the animals rejected their graft, or you deplete these cells in fast ligand engineered and long-term graft recipients, and those graft recipients will actually reject the graft. So demonstrating that in these T regulatory cells are critical for the tolerance that we observe. Now then we ask the question is that, do we see higher frequency of these T regulatory cells in long-lasting animals or long-surviving animals? And we look at use flow cytometry. When you look at the cells, really, especially the kidney draining lymph node, remember what as I showed in the previous slide, the action takes place in kidney draining lymph node. More or less, we got the same number of FOXP3 positive T regulatory cells. And as it's shown here, we didn't really see, especially kidney draining lymph node, long-term animals did not have higher level of T regulatory cells within the kidney draining lymph node. All right, so then we asked the question, there are two possibilities. Number one, maybe they are actually within the islet graft themselves, or it's really it's not the matter of the number, it's the matter of function. So this is what we wanted to prove it. And when we look at using confocal microscopy, when we look at the T regulatory cell within the graft, yes, indeed, we've shown that fast ligand engineered long-term grafts, and this is over 200 days, they have more green FOXP3 cells as compared to the controls. I'm sorry, this is day 5, I apologize. So we also look at long-term, but this is day 5, this time kinetics, we see more T regulatory cells within the graft. And we also saw higher level of macrophages, and importantly, macrophages that are expressing TGF-beta. I'll tell you the significance of that in a minute. So when we tabulated the number of T regulatory cells infiltrating in allogeneic pancreatic islets at day 5, and yes, indeed, you see a higher number of these T regulatory cells within the islet graft itself. So then we asked the question is that, can we do adaptive transfer experiment to demonstrate that indeed these T regulatory cells are relevant? So we get the T regulatory cells from kidney draining lymph nodes, and then we adaptively transfer. In this study, we used between 2,000 to 10,000 T regulatory cells and 1 million conventional T cells. We put into a rag knockout. It's actually an OT1 transgenic mice. The bottom line is that these mice all they carry CD8 cells, and they express all those CD8 cells. They express T cell receptors specific of albumin, as I indicated before. These animals will not reject biopsy islets as is shown here. So they are incompetent because they only express a restricted CD8 receptor that is specific of albumin. When you transplant 10 million CD4 cells, if you notice, these animals will reject the islet. And if you take naive T regulatory cells with 10 million conventional T cells, the naive T regulatory cells will not prevent the rejection. But if you take T regulatory cells from tolerant animals, and they will actually prevent rejection. And we only, in some cases, we only use 2,000 T regulatory cells from long-term animals. So the other thing that I didn't show here, if you take CD4 positive, FOXP3 positive T regulatory cells from the spleen of these animals, they actually could not protect. Okay, there is slight prolongation indicating that those cells were not antigen specific. So we think what happens is that these are antigen specific T cells within the graft draining lymph nodes, and therefore you could use as many as 2,000 to protect the rejection from 1 million effectors. And then if you remember, I told you, in the graft we saw a lot of macrophages, so that we asked the question, what is the role of macrophages in this model? We use clotrinate to eliminate those cells, and this is just to tell you that yes, indeed, when we use clotrinate, we eliminate both macrophages as well as immature dendritic cells. And interesting enough, if you deplete phagocytes at the induction phase, our tolerance regimen was actually ineffective. So these are the animal receiving fast ligand engineered islets. They survive, they don't reject. And if you deplete phagocytes, all those animals actually rejected. And then I also told you that we saw macrophages expressing TGF-beta, and this is a classic picture that happens. So we asked the question, is TGF-beta is playing a role? Indeed, yes. When we block TGF-beta at the induction phase, not at maintenance phase, we could not induce tolerance. And blocking IL-10 did not have a major impact on the survival. So we also shown, in vitro, if you take simply, we've taken our transgenic system, OT1, and we co-incubated them with macrophages in the presence of fast ligand, and we've demonstrated that macrophages will take apoptotic bodies shown in green. These are alloreactive T cells, and in the process, they will make TGF-beta shown in purple. So the other thing that we've demonstrated that tolerance is localized because these animals, they had a normal donor response in the periphery. Then we asked the question, is it possible that tolerance is actually localized to the graft itself? And to provide evidence for that, so what we did, we induced tolerance with fast ligand engineered islets, and then at day 100, we remove the original graft, and then we transplanted these animals with an unmanipulated second set of islet under the contralateral kidney capsule, the other kidney capsule, and at day 100, all these animals actually rejected the second set of graft. And then we thought maybe is it possible this has to do with inflammation, surgery-associated inflammation, and what we did not remove the primary graft. We used an unmanipulated second set of graft put under the other kidney capsule, and 40 days later, we basically removed the kidney that had the original graft, so that we wanted to test the effect of inflammation. When you remove the primary graft, if you notice these animals develop hyperglycemia, indicating that the second set of kidney had already been rejected. So definitely the tolerance is localized to the islet itself. And we wanted to see, is this tolerance tissue specific or not? And to prove that, we used donor skins, biopsy skins at day 100, we put onto those animals. The skin rejected, but in the process, islet rejected as well. So when we put islet graft, unmanipulated islet graft, under the contralateral kidney capsule, those grafts will reject, but that rejection reaction did not precipitate the rejection of original graft. But you do the skin, actually you also eliminate original graft. What we think is happening here is that because skin generates such a potent alloreactive response, maybe it's a balance between your T effector and your T regulatory cells. In the case of islets, you're not generating so many T effector cells, therefore they're not sufficient to overcome the T regulatory cells that home to the original, the primary graft. Okay, so we are in the process of actually testing that concept. So we think this is what's going on in the system. So we take pancreatic islets expressing Fas ligand. The pathogenic T effector cells, they try to reject the islets. In the process, they receive the Fas signaling. They undergo apoptosis, and phosphatidylserine is being put on the other portion of the membrane, being recognized by phagocytes. Phagocytes chew up basically these apoptotic cells, and in the process they make TGF beta, and TGF beta is well known to be involved in not only the generation of T regulatory cells but also improving their function. So, and these T regulatory cells, we think they go to the pancreatic islets, they home to the islets, and they locally protect the islets from T effector cells. So these are the conclusion. So immunomodulation with Fas ligand engineered islets result in localized tolerance. Tolerance involves the clonal deletion of alloreactive T cells, and that also initiate a complex immunoregulatory loop that involves phagocyte, TGF beta, and Treg axis. Tregs are involved in induction as well as protection, long-term maintenance of the graft. So these are my colleagues. They did the work, and I appreciate the support, from various funding agencies. With that, I'll be happy to take any questions you may have.
Are there any questions?
Paul.
Any PD-L1 production by islet cells?
We didn't look at. I mean, that's certainly something we need to look into it. We know that PD-L1 it is important, at least in NOD setting, it is important for maintaining autotolerance, because Mosaic and colleague, years back at Harvard, they've demonstrated if you knock, PD-1 and PD-L1 in NOD mice, the diabetes, there is an accelerated incidence, of diabetes, in those animals, demonstrating that PD-L1 is really critical. I think Paul, that's a great question. We all know what PD-1, PD-L1 system has done in cancer setting, right? I mean, a lot of blocking antibodies now approved by FDA and all that. I think it's a very robust system. I'm not presenting the data, but we currently have funding from NIH in collaboration with Georgia Tech. Actually, we're using PD-L1 for exactly the same purpose. Very interesting molecule. Other questions?
Have you patented this process? Up to patent?
Yes, we have over a dozen of patents, on this concept, the platform concept. We have actually worldwide, protection, on this, right? I think we filed close to 35 patents. We have a dozen approved here in the US, in Europe, and other places. Yeah.
Are there any further questions?
I think I just want to make another point. I'm sorry, I'm not showing the data, this is really a very exciting technology, and it has application, particularly in stem cells. We've shown you could take hematopoietic stem cells by putting Fas ligand on the surface of those cells. You could overcome rejection, and we have demonstrated that you could use this concept under non-myeloablative conditions. You could do bone marrow transplantation, and you can avoid graft versus host disease because Fas ligand uses the same loop. So we're very excited about. I'm not talking about it, but we have a program actually leading, that effort. Hematopoietic stem cells and maybe potentially some other mesenchymal stem cells, etc. Thank you.